doing here. Oh, sorry. So today I will introduce you what I'm working on. This is today's my presentation. The advances in the systematic of Extant Myrmeleontidae, aka the antlion and onflies. First, sorry. First I introduce my name is Yuchen Zheng. I'm from China Agricultural University, and my supervisor is Xingyu Liu. And my host, and on email, he hosting me here, so it's my supervisor. And my research area is the systematics of the superfamily Myrmeleontidae. My PhD project is systematic of the two subfamily within the Myrmeleontidae, the Dendroleontidae and Myrmeleontidae, focusing on the Oriental region. What is the Myrmeleontidae? It's a large family in Neuroptera. It's holometamorphous. How to distinguish them? First, it has a clubbed or spoon-like antenna. It looks like the dragonfly, and it has two pairs of membrane wings with complex venation, and it has its adult has elongated abdomen. And its larva is all very well known. There are many trap building species, and its larva has a well-developed curved toothed, grasping, sucking mouth parts. There is some synapomorphism for this family, like there is some cross veins between the vein RA and MA, and its larva abdominal segment 5, segment 9 has some digging city, has some robust digging city. It's not like other lacewings. The other lacewing has adaptive picopod. Picopod is pink. It's pink like the green lacewing, So the systematic position of the Myrmeleontidae. First, introduce the Neuropteridae. It's a superorder sister with the Colepteridae, and the Myrmeleontidae is a crown group. It's a crown group of the Neuroptera. But everyone knows the insects is the most diverse. It is the most diverse group in animals. Nearly 100 million species. Neuroptera is a small group. It's a very small group, very small order. Only 5,500 species. But Myrmeleontidae, there are one third within this group. So they are the most diverse family within the order. So many species, but for now I introduce the first described antlions and onfly. As we know, previously the traditional classification, they are separate family, but for now they combine the same family. The first described antlion is Myrmeleon formitarius, very common in Europe. Also, the Libeloides macaronius widespread in Central Europe to China. So the traditional classification of the Myrmeleontidae and Ascalaphidae is only based on some subtle characters. For example, such as Ascalaphidae, the onflies, they used to be treated in three subfamilies, like the Haplogrini is an entire eye group. With a normal eye, and elongate antenna, elongate butterfly-like antenna. And Ascalaphidae, this is a group with its eye has a split split change. Has a split change with its eye is split, is divided, and with also with elongate antenna. And the other and the last subfamily is Arbardinae. It's a monotypic subfamily from Brazil. Only contains Arbardina furcata with short club antenna and a normal eye. And a normal eye. And the Myrmeleontidae, it also contains 3 subfamilies and many tribes. And you can see there are still some transitional group between these families, both families. And the previous researchers, they divided them only based on some subtle variations and a single character like the hypostigmatic cell under the peristigma. They said the antlion has an elongated hypostigmatic cell, and on the own flies, the escalaphid has short, but there are always some exceptional. There's some people, they reconstruct the phylogeny for the entire Escalaphidae and Myrmeleontidae. It's not surprised the own flies are deeply nested within the Myrmeleontidae. You can see the green group is the previous own flies. They're all nested within some antlions like the Paparinae antlions, the very glorious large group. To make people to understand why there are so complex systematics, I think I should present you some of the representative diversity of this family. First, Myrmeleontidae is a worldwide group. It's cosmopolitan, but in general and triple level, they are mostly they are endemic to single regions, or in single regions or the neighbor regions. They are more endemism in the generic level. Body size, some of them are very large, some of them are very small, some of them are very small. Also, they are diverse in their antenna. The typical on fly, they have elongate butterfly-like antenna, and some of them has a field form antenna. Very atypical, and some of them has a thick antenna but not swollen. And they also have a diverse leg, even in the same subfamily. Even in the same subfamily, some of them are elongate, some of them are short, and within different subfamily, different tribes, they present some similar shape of the leg, some robust leg, or you can see some group even involve some spine, like some mantis. They involve some spine, and like some of them also elongate elongate the tassels for climb or for protect. And everyone focusing on the Neuroptera, they mostly firstly focusing on their wings, so like some the complex variations and the diverse of the wings markings. We can find many glorious species in every zoological region. Even in China, we have many beautiful species. And worldwide there are also many beautiful groups like this Cynosalis ploveri from Madagascar and some species from Africa. Even some of them have a very unique colors. Some of them is pink. It's pink and has a eye spot on its wings. Or some species has a metallic blue on its wings. or they have some minimize for leaking. And this species, I think, many of you, maybe sometimes when you clear, you can find them. It's very common in Mediterranean area, especially in south of France. And they are very diverse in their sexual dimorphism. Normally, the species normally the species has normal. There are not so many different between the male or female. Only different with the genitalia. But there are some male. They involve some different character, like an elongate ectoprop to enhance to mating for mating, or it's presenting different group. And some of the male its antennal, the shape of the antennal is different with the female. You can see the male has a more curved antenna. Some of the species has extremely elongate ectoprop, such as the Paparidius capicola from Southern Africa. There is not only elongate for its ectoprop, also presents some elongation for its abdomen, It's also present in different groups. You can see they are from different subfamilies. There are some unique diagnosis on the male, like the pilula axillaris is a unique character in this structure is for controlling spread of pheromones. It's not for spread the pheromone. It control to spread the pheromones. When the wings folded, this structure can stop the hole on the basal abdomen to controlling spreading the pheromones. But for now, we don't know the function of the pheromones. Maybe it's related to for mating, but we still don't know now. And the sexual dimorphism is not only present on the elongation, also the shape, present on the shape. Like the Asian genus Epicantha crisis, is many males has a swollen and modified abdomen. And this modified abdomen sometimes spreads some mucus. It's also an unknown function. Maybe it's to attracting the female. Maybe it's used for feeding females. We don't know. We still need to verify. There are also some of the species like Nophis from Morocco. Its male abdomen is curved and empty. Still mysterious function. And all insectile group, we divide the species. We mostly focusing on its genitalia. We all know the male genitalia is important to divide the different group. But in Myrmeleontidae, its female genitalia is also very important to differentiate the different group. You can see it reflects this genitalia reflect its ovipositing behavior and the larva habitat. Like if there is a group like some onflies and some Dendroleontinae lions, the larva lives on the tree, lives in the tree hole or some place without sand. Its female genitalia the sepal is slender, not modified. But for the group for the group lay eggs in the sand, the larva lives in the sand. The city on its female abdomen usually modify like specialize some six city for digging the sand to enhance their ovipositor. You can see I already marked the larva habitat. So why the antlions and onflies are so diverse within the Neuroptera? In Neuroptera, it's because they always colonize the different habits, colonize different environmental niching. Even in a related group, their habitats are different, such as the Dimarini from. For now, I introduce you some representative larvae in all subfamilies. For some subfamilies, such as the first is the Ascalaphini. It's a Ascalaphini. Including the Paparini, antlions, and the onfly, such as the South American Dimarini. They live in the deep sand, maybe 40 centimeters. So you can see their mandibles, their head are shorter. They don't need to ambush on the top of the sand. So they only need is dig and find some arthropods from the deep sand. But you can see the Paparini, they have developed mandibles. They can live. You can find the different species in different habitats, like the Paparilius phosphoi. They live in the coastal beach, and the Paparilius libeloides. You can find it in the thick dry soil in the Mediterranean area. And Coreophrus merely is from the dune of the inland of the southern Africa. And the onflies, mostly they are naked. They not bury themselves within the sand. But there are some exceptions, like the Spheropterygids. They live in the sand. But most of them they live on the tree, live on the dead leaves, live on the rock surface. And you can see they have developed projections on their lateral body. We call it Scoli or setiferous process. Please remember this character. It's very important to understand their behavior. I will present you later. The Myrmeleontidae is the most well-known group within this family because they make the trap. There is a public misunderstanding. Many people think that all the antlions they make a trap, but in fact there are only 20%, nearly 20% species they make a trap. Now others are ambushers and they don't make the traps. All trap make, including in these subfamilies, and they are related, but not monophyletic. They are related but not monophyletic. So these three species you can find the single island, the island I live in China. Like they also they make the trap. They separate their ecological niche. Like the Myrmeleon exiguus, they live on the coastal beach. And Myrmeleon tenuipenis, they live in the mountainous sands. The Creta cruzi they live in the open area. They all live in the open area, and their trap are modified because they not only make the trap, they also make the channel under the trap. you can avoid the sun because to make the temperature getting lower because they live in the open area. It's hotter. It's hotter. And some of the species also modified. Is a highly modified. Has a highly modified trap like the Australian Callistoleon species. They make the pits like the star. They expand their trap. They make many trench. They make many trenches. And as I said, not all the Myrmeleontidae they make the trap. There are some species they have the other behavior like the synclisis. They are active. They are active. They are active. They are active predator. You can find it in the coastal area from Europe. And the Brachynebulosa from USA, they minimize the motility of the Hymenoptera, Hymenoptera, and the Neolatus, they live in a specific plants, puya, to protect the some butterflies or some beetles that to be attract these flowers. And many also there's still many species looks like the on-flies larva and lives on the exposed area. There are some species lives in the desert, lives in the rock of the desert. And not they are all of them are not trap maker. And Dendroleontinae is a group I'm mostly focusing. It's diverse in Asia. It's diverse in Asia and Australia. You can still find in the same group in the same group. Some of them live in the sand. Some of them you can find it on them exposed and on the cast stone or on the stone with dust and leaching and some of them they bury on the thin sand on they bury with some thin sand on the rock surface and you can see they have some city to cover many debris to attract the prey to feed and then capture them and the last subfamily is Myrmeleontidae. They also those species are also found in the same island I live. Some of them still they separate their environmental chain, such as like you can find the Distoleon species from the mountainous sand and Paragranulus pumilus are endemic in the roots of the coastal plants, and some of them you can find in the artificial environment. And you can still find some Ascalapid-like species within this group. So if we mark all the character, all the species, all the representative species, you can find there are many characters are irregular in the phylogenetic trees, and some of the character is useful on a generic level, but for triple level for the subfamily level is useless. It's relatively useless because there are some characters are evolutionary adaptations. They are plastic. So as I told you, the scoli, the projection on the lateral body is not only present on the antlions, also found in some Chrysopidae species. And you can see if the species lives in the sand, they are terrestrial. The scoli are absent. So the skull also found. You can see the nymphed species is a family related to the Myrmeleontidae. there is a sister group. There one subfamily has a elongate story, only live on the tree, live on the exposed area, and the species which lacks the story, they live in the sand. So the story is for sometimes the researcher consider is for camouflage, or the stabilize for hunting. So when they live in the sand, the story will be weakly developed. So it reflects a question. If we reconstruct for the fossil species, especially the larva, these such characters it's very difficult to reflect the logical phylogeny for the trees. So how to and there's a many topic about how the antlions and onflies involve so diverse ecological linkage. They are origin from the Semophilous or the arboreal. We still don't know. We need to reconstruct the logical phylogeny for this family. For now, there are many results for its phylogeny. The present acceptable result is the Machado et al. 2019. They used the HE database. And some of the very large data set that only include a few samples, so cannot reflect the entire relationship within this family. We cannot ignore the geographic isolation because we all know to make a group be diverse, it's not only depends on the diverse of the ecological link, also the isolations. So as I told you, that the group within the Myrmeleontidae, they are mostly endemic in the same regions or the neighbor regions. And it reflects another question. There are many species, even they are not really related in the molecular phylogeny. They are from different zoological regions, but they have a similar patterns on their wings. Also, the shape of the leg of the bodies. So in the taxonomic history, there are many shortcoming of the issues there are many species are including the same genus only based on the wing marking, but they are not related. It's a parallel evolution. But in fact, the Myrmeleontidae is a very good material for biogeography research because they are worldwide group, and most of the species have poor dispersal ability, and they are highly dependent on specific habitats, and they occupy diverse ecological linkages and contains many widely distributed high-level lineages across the regions. And there is few invasive species. So there's not. So it's very nice to make the biogeographic analysis for them. But there is a challenge. Most of the lineages, they are monophyly remains unverified, and the accuracy of records for many common and widely distributed species is questionable. And the larva study is only focusing on the pit building species, on the trap building species. And many countries and regions still lack the comprehensive diversity investigations. So, for example, there are some example for the chaotic systematic within this family, such as the tribe Myrmeleontidae contains the type genus, there are many results for their phylogeny. Also there is a result I haven't published. You can see all the Myrmeleon species. It's not monophyletic. There are many genus. There are many varied genus are deeply nested within the Myrmeleon, and you can see they're from everywhere. The genus Myrmeleon is now a worldwide genus, which is not really logical. So the type species of Myrmeleon is from Europe. It's from Europe. Myrmeleon formicarius, I've mentioned before. And previous researchers they mostly treat the species within the Myrmeleon only based on some venations, the markingless characters, and there are also some exceptions, like the Myrmeleon eurycephalius has many spots, and they don't, and the previous researchers they mostly don't focusing on the genitalia. But if we dissect them, their genitalia is very different. It's very different. You cannot find the synapomorphosis among them. So based on my present molecular phylogeny, the UCE based, we can find the Myrmeleon formicarius group. This group from Europe, the real Myrmeleon is sister with a valid genus Hagenomia tristis from Africa. And they are very different on the morphology. Not only the single character. There are many differences on the venations, the shape of the wings, the bodies, and the genitalia. And the most important is the rare Myrmeleon formicarius lacking the pilula axillaris I mentioned before. And other species, other group, they present such characters. And also reflect on the differentiation of the female genitalia. You can see like Anomia had a very elongate gonophysis on its genitalia. And if we can see from my tree the many valid genera is totally nested within it, and their characters are for the first glance is very different. It's very different, and we can see the Myrmeleon species. The genus Myrmeleon has a large number of the synonyms from the taxonomy history. So it needs a very big revision. For it. For example, the real Myrmeleon. As I told you, the real Myrmeleon is only found in the Palearctic. I summarized the diagnosis. There's a similar shape of the male genitalia, and they are females genitalia are all the same, and all of them are lacking the pilula axillaris on their hind wings, and also the similar venations, and they are markingless. There's not only a problem on the genus Myrmeleon. There are still some issues within the type species of Myrmeleon formicarius. For now, their record they record the entire Paleolithic region from Spain to Japan. But in fact, the Myrmeleon formicarius is only found in Europe. It's only found in Europe, and the species from China, from Japan is a different species. But now they're all synonym. They're all synonym. For now, my result is based on the morphology. But when I go back to China, I will do more molecular evidence. So based on my research, I summarize all the valid genera and give a list for all the synonym which needs the future revisions. The other example is another European species, the Dendroleon panzerinus. Dendroleon panzerinus is widespread in Europe. Widespread in Europe, and it's not so common. It's not so common. It's relatively rare. So there is a complex. It's a species complex. The Dendroleon panzerinus is found in the type locality is from Austria, and the Dendroleon simile is very similar to Dendroleon panzerinus. They are from the type locality is from north of China. It's from north of China, so you can see they are very similar on their wings markings for the first glance, but both species is now recorded in China. It's all recorded in China. They're all recorded in China, and if we check their distribution, there is a large distribution gap between this species complex. As I said, the type locality, one species type locality is from Austria, one is from the North China, and they're indeed very similar on their wings markings. But after I dissect them, their genitalia is relatively different. Their body markings also there are some different in the species level. But the most important is there is obviously differences between the larvae. The larva of the Dendroleon panzerinus it has a tooth of a city on the mesothorax, but it's absent on the Chinese species. And you see there are different marking, different colorations. And I also do some molecular for them. There's also still distinct gaps between the European species and the Chinese species. So the conclusion for me now is the Dendroleon similis and Dendroleon panzerinus is a two independent species, and the distribution records of Dendroleon panzerinus in Asia is a misidentification of the Dendroleon similis, and they have very clear geological isolations. And this complex has more species, in fact, like the species from the Iberian, from Spain, from south of Spain. Previously, it was treated as tricholeon. The tricholeon used to be a endemic species, endemic genus in South Africa. So it's not really logical. There is a species found in Europe. And the researchers, they treat it as a tricholeon based on the wing markings and the body markings, but you can see their body properties and it's different. I also dissect the genitalia on the PPT. Sorry, it's also very different. In the tricholeon relictus from Spain is more related to the Dendroleon panzerinus morphologically, and also verified from my phylogeny. From the phylogeny, you can see the tricholeon relictus is native within the Dendroleon panzerinus group. So it should be a new combinations. And we also found some new species from the museum, and based on my collecting, one new species from the south of Anatolia, one new species from the Qingling Mountains from China. And the third example is the tribe Megistopini. Based on the phylogeny tree of Machado 2019, they're only including one species. Other genus were including this tribe he designed is based on some morphological results. So I want to verify the monophyletic of this tribe, and for now all the Asian species are included in this tribe, Myrmeleontidae, but there is no molecular evidence. So I included all the morphological related species and genera in my molecular phylogeny. The result is very satisfied. The Megistopini is very diverse in Asia. It's very diverse in Asia. And the genera the Machado he designed from the Europe is also nested within it. So he's correct. And we also do some biological analyze. This tribe was originated from the Himalaya to Zagros and spread to Europe and Southeast China. And you can see they're also different in their different on the morphology. The end genitalia is a Myrmeleontidae genitalia. It's a genitalia of the Myrmeleontidae. It's very typical. The GX9 fused as a fork shape, and you can see all other Megistopini species. They have their GX9 like a pair of plates. So there are some symptomorphisms within this tribe. And reveal more questions. What's the systematic position of the closely related group from Madagascar? From Madagascar and to Seychelles. This fauna has some very morphological related species with the Eurasian. From the Eurasian, you can see they are very similar. The body shape, the wing markings, the legs shape, and the genitalia, like this one. This one is from Madagascar. This one is from Madagascar, and this one, and they are from, and G, H, I, and I. They're from Eurasia. So still some question need to be verified in the future. And so there are three examples for the chaotic systematic problem of these families. And I also in the past few years I also made some research there are some research progress for me. For the past 5 years I mostly focusing on the Asian antillions revisions, especially some very puzzling group like the sister group Gazala and Nexarus. It used to be a group. You can see there is two types of this group. There used to be three in a single genus Gazala, and there is another monotypic species genus Nepsalus, only contains a Nepsalus indicus, but we can see they are from the same geological region, and they have the same markings and shape of the wings, the body size, and the leg shape is the same. So I do the molecular phylogeny and morphological examinations. For now, I transfer most of the species placed in Gazala Nepsalus. The Nepsalus is not monotypic genus. In fact, Gazala is only including two species. Its wing marking is relatively scattered, and it's not. And the differences between them is not only reflect on the wings markings, also reflect on the genitalia. There are some synapomorphies, like the Gazala has a tiny PGXA, and in Nepsalus is longer and distinct. You can see the GPA is in Gazala is more scleralized, and in Nepsalus is more membrane. Also found in the differences from the larva. The Gazala has a pale body, and all the Nepsalus species has green colorations. But they are ecological link. The ecological link is the same. All of them lives on the rock surface with the dust licking. But how to say but Gazala is only found in the mid elevation of Himalayas. But the Nepsalus, they can find entire mountainous, moist area of the East Asia and Southeast Asia. We still do the biogeography of for them. Is Himalaya origin, and still we also find some new species, one in China, one in Sri Lanka. For now, in my conclusion, Gazala only found two species in the Himalayas, and Nepsalus is widespread in Asia. And there are still more new species I haven't published, like this species has a transition character between Nepsalus and Gazala. Maybe in the future they should be combined in the same genus. We don't know. Maybe I need to do the analysis in the future. And my another progress is for the tribe Acanthoplextrini. Based on my analysis, the diversity of this tribe before me, there is only 8 species in Oriental region, but for now there are 16 species. We also found many larvae records. The larva is very important for the analysis of the ant lion because they reflect many behavior and characters. Within this tribe, I also make a revision for the Asian genus Extant species. Previously 11 species, but for now there are 22 species. And still many larvae records are found. Many larvae are recorded. To do this is for the more comprehensive phylogeny for group. So after my revision, I want to combine all the results for the entire phylogeny for these subfamilies of the Neuroptera. But this tree still need supplement. I need to add more samples from Madagascar, Southeast Asia, and Africa. But my sampling is very satisfied. I will do it when I'm back. Everything we need type examinations. Without the type examinations, every result is not stable. Every result is not stable. We need to improve our knowledge. So that's why I come here for a year to examine numerous types. Is for accuracy of my knowledge, and maybe in the future the data set can be published with the curators. It takes a time because it's not good for only photo graph the habitus, I still need to dissect them. There are many information in their genitalia. All of this is for a very big project for me is evolutionary history of Myrmeleontidae. For now, I already sampled more than 400 species, but the number is still increasing. Maybe in the end, I can sample maybe 700 species, but who knows? Who knows? The other of my progress is the progress is a monograph of Chinese Myrmeleontidae. We now found many new species and new records in China. Nearly 100 species, but for now, we found new species based on my collecting, based on my social exchange. There are many new species, new records, and unpublished data. For now, 220 species, but the number is still increasing. To all this, I want to say thanks for all the people supporting me, assist me in Europe. First is Xingyue Liu. He hosting me here. He helped me a lot. Also, there are many curators from other countries, Ulrich and Horst Asko, Susanna Randolph, Davide Badano, Peter Merchelik. They assist me to examine many European species and types. Also in France, Bruno Michel and Labrique, and Fabian Condamine, they give me many advices and let me examine many of their collections. Also, the people in the museum, Quarantine and Anchen, Yu Zeyuan and Li Lingwei, Mathieu. And Fred, we did Fred to take photos, and Valerie Pullman, they also helped me a lot. And thanks to everyone. Merci beaucoup. That's my presentations. Thank you.
Thanks.
Thank you.
Your speech and your huge work and beautiful pictures.
Thank you.
You have so much thing in progress.
It's in progress.
Yes. Okay. Do you have any question for our discussion? Yeah. Oh, yeah.
So I'm a doctorant in this group, but given that the larvae, many species that are there are living in cells.
Yeah.
So do you know if in the deserts there are diverse species?
Yeah. They are very diverse in the desert. They are very, very diverse in the desert. But in the mountainous, they are also very diverse. It's also very diverse, but it's more difficult to be found in the forest. Now on open area in the desert, in open area we just put the light, and every species can come from the light. But in the mountainous, even they are diverse, the forest, the trees will avoid the light. There are many shades, so the interlight from the forest is difficult to be attracted. But in forest, they are still diverse. But most diverse is in desert. They are most diverse in the desert. In dunes, yeah, they can find everywhere. Maybe next to the building, there are some interlights.
I have a question about the preservation of the larva from the field. How can you collect the larva and preserve it dry, alcohol?
When I collect I collect them by some shelf. How to say that? By some shelf, by some equipment. I don't know how to say that, but you use something to dig them, or sometimes I use my eye. Find the rock servants, or use my hand to touch the touch the or brush, touch the thin sand, touch the thin sand. If there's a soft things, maybe it's a lava.
Yeah.
After I collect them, there are two options. One is I rear them to the adults.
Oh.
But mostly if I collect less lava and I cannot identify them and they are rare, I prefer to keep the specimen in lava stage because the lava morphology is very, very important. And I identify them based on the molecular, based on the barcoding, based on the code barcoding to match the adult, to match the adult. So sometimes I rear them, but sometimes I put them in the alcohol.
No, in alcohol.
Yeah.
Okay. There is no dry collection.
There's no dry collections for the lava because if we getting dry the lava, the condition of the lava specimen is very bad. Yeah.
I don't know if you have some question online. Sorry.
You mentioned that you were using the UCD data, right?
Yeah.
And how many loci are you thinking about? How many loci?
Loci. Nearly. It depends. Mostly, I can get 3,000 loci. 2,000 to 3,000 loci. I think it's enough.
Yeah.
In fact, more than 1,000 is enough. I usually can get the 3,000 loci. But it depends on the condition for the samples. It's better for the recent specimen preserve in the alcohol or dry immediately and put them in the fridge. Yeah.
And did you generate the base for the data or was it already available?
Sorry?
To sequence this loci,
yeah,
you need to target them.
Yeah, I need to target.
Did you have the data already?
Yeah, I already have the data. I designed a probe myself.
You did? Okay.
Yeah, I designed a probe myself. Yeah.
And did you try to sequence some museum specimens for the UCI? Museum specimen. Did you try to sequence museum specimen for the UCI?
No,
it's only
museum specimen is very difficult, and if they are spread wing is very, very difficult to get the data. Maybe it can only get the code 1 data. But the specimen put in the alcohol and the recent specimen, maybe 10 years, the data is very satisfied. But after if you prepare them, put them in the water, the DNA condition is getting worse. So it's better to sequence the specimen in the alcohol or unspread specimens.
On peut s'arrêter là. Merci à tout le monde en tout cas.
Merci.
Au revoir.
Merci.